Yeah, can you see my screen now? Yeah. Uh, yeah. Good afternoon, everyone. I'm Gaurav. Uh, we are from BITS Pilani Hyderabad campus. Today, here are presenting. I'm Gaurav, Abhinav, and Abhishek under the supervision of Professor Shankar Ganesh sir.
Hello, I'm Abhishek. I'm gonna introduce the topic. Municipal wastewater is a common form of low-strength waste stream that contains organic matter and pollutants. And the municipal wastewater treatment facilities have the potential to become net producers of renewable energy by transforming the organic contaminants into biogas. However, household products like medications and detergents can cause the micropollutants to be released into the environment. So it is important to understand the micropollutant behavior during wastewater treatment process to prevent the environmental damage. So overall, the municipal wastewater treatment plays a very critical role in protecting the environment and even producing the renewable energy.
I'll take on from here. Uh, Uh, Coming to antimicrobial resistance, it is a global public health crisis that occurs when bacteria, viruses, fungi, or any other parasites develop resistance to antimicrobial drugs. AMR can be acquired through horizontal gene transfer and is caused by quorum sensing of microbial population, which regulates gene expression and allows bacteria to combine and resist antimicrobial medications, disrupting bacterial cell communication, which is one way to minimize AMR. Biofilm formation. Microorganisms such as bacteria often attach and grow on surfaces, which can include medical equipment, living tissues, soil, and water. They produce a sticky layer called biofilm that helps them stick to the surfaces and protect them from antimicrobial treatments. Antibiotic concentrations required to destroy these biofilms are about 10 to 1000 times higher than those required to kill free living cells. And biofilms contain antibiotic resistant cells. These factors contribute to biofilm medicated antimicrobial resistance, making it a challenge to treat device related infections. Algae as anti-biofilm agent. Microalgae found in freshwater aquatic settings contain bioactive molecules that block quorum sensing in various bacterial species, including gram-negative bacteria. The bioactive compounds extracted from microalgae can treat both types of autoinducers that are used by gram-positive and gram-negative for cellular signaling. These compounds have potential to inhibit the formation of biofilms and eliminate bacterial colonies on biotic and abiotic surfaces. Despite the presence of several antimicrobial compounds in microalgae, there is a comparatively little research on their antibiofilm activity. Further research has revealed that brominated pheromones can operate as potent inhibitors of quorum sensing mechanisms in both gram-positive and gram-negative bacterial species. Aims and objectives of this study are to identify the role of algae as antibiofilm agents and as agents for antibiotic removal, to study the impact of algae on the biofilm using bacterial biomass, to understand the effect of antibiofilm and quorum sensing inhibiting agents and preventing antimicrobial resistance. From here, Abhinav will take on.
Yeah, I'm Abhinav and I'll be taking over from here. So this is the this is slide containing about MBBR, which is moving bed biofilm reactor. So MBBR is a wastewater treatment process that uses a suspended carrier material to support the growth of biofilm. Microorganisms attach to the biofilm and treat the wastewater as it flows through the reactor. So the carrier material moves freely, creating turbulence and improving contact between the microorganisms and wastewater. So basically, MBBR is an efficient and problem method for biological wastewater treatment. Next. So let me uh, show you the flow of project. Basically, we have set up a lab scale MBBR system and then cultivated 3 microalgae, which we'll be looking at. Then the small scale study on effect of single and multiple combinations of algae on biofilm was done, and then incorporation of the best outcome from the above combination is set into the MBBR system, and the final inference on algae against antimicrobial resistance. Next slide. So the lab scale MBBR setup. The lab size system is a scaled down version of the larger 600 kiloliter per day MBBR system and is designed to contain 2 portions of treatment. So one is for testing and the other is for the control. To verify the accuracy of the calculations, the input characteristics of the larger system were subjected to the calculation techniques which were outlined in the book Wastewater Engineering by Metcalfe and Eddy. So the same methodology was then applied to calculate the treatment capacity of the lab scale plant, which was set at 50 liters per day capacity. Next slide. Cultivation of microalgae. So to cultivate the 3 microalgae which we choose are respectively Chlorella sorokiniana, Necloris oleoabundans, and Nanochloropsis oculata. So these algae were first grown in 250 ml of BG11 media. Later, the culture volume was increased to 700 ml. So the microalgae cultures were maintained in an orbital shaker incubator at 26 degrees centigrade temperature and at 110 RPM. Also, 2 LED bulbs were used to provide adequate light for their growth. Next, test for presence of antibiotics. The raw wastewater was first concentrated, which was first concentrated at 1000 ml, was decreased to 100 ml in a hot air oven to increase the concentration of antibiotics, which are usually present in a very low amount. Nutrient agar plates were then prepared, which were streaked with E. coli strain of optical density 0.7. So 3 holes were punched in 2 of the agar plates, and one of the agar plate was inoculated with 100 microliter concentrated wastewater in all the 3 holes, while the other plate contained 200 microliters of wastewater. So this was done to test the effect of different concentrations of wastewater on bacterial growth inhibition. And also for reference, 3 other plates were used by placing antibiotic discs such as penicillin, erythromycin, azithromycin, and ampicillin, etc. Next. So upon this test for presence of antibiotics, it was observed that there was no zone of inhibition in the plates containing the wastewater, which basically indicated that we needed to use some other advanced technique to analyze the antibiotics present. Next slide. So this is what we have done till now, and the plan of action for the rest of the project will be to firstly estimate the antibiotics present in the water. So the solid phase extraction is to be done for the extraction of antibiotics from the wastewater, and the concentration can be estimated using tandem mass spectroscopy equipped with high performance liquid chromatography LCMS using C18 column. Also, the test for presence or absence of biofilm is to be done. Wet and dried algal biomass are to be collected separately, and different combinations of these algae are to be incorporated into the wastewater. So we'll choose the best combination from these combinations, and then this can be introduced into the larger scale MBBR system. Biomass quantification can be done using the SILE method for biomass quantification. Thank you.
If anyone have any questions, you can ask.
It was a good presentation. Thank you for your presentation. Still, we have only few references, right, sir? So, can you please share your references with us?
Hello. So thank you for your presentation. That was a nice presentation. Can you please share your references with us?
Sure, definitely. We'll share the references. We'll send it to the mail ID that was shared with us.
Thank you, sir.
Thank you, Gaurav team, for this paper presentation. Next, I would like to call upon Harshvardhani, online presenter, to present her paperwork. So I can't hear anything. ஹர்ஷவர்தினி இந்த மீட்டிங்ல இல்ல, நாங்க எந்த மீட்டிங்ல இருந்து நாங்க கேட்போம். Until the presenter comes, we will be waiting. இதெல்லாம் ஓப்பன் பண்ணி வச்சிருக்கோம். நல்லா அட்மிட் பண்ண போறேன். Yeah, okay. Harshavardhan, can you audible me? Anyone can audible me? Hello. Am I audible?
Yes, sir.
Yeah, okay. First, I would like to welcome Ms. Harshavardhani to present your paperwork. You can start. Sure, okay. Now, Rajkumar, present your paperwork.
Okay, sir.
Start presenting yourself and then you can present.
Okay. So shall I start? Can I start?
Yeah, okay, you can start.
Okay. Good afternoon, everyone. First of all, I'd like to thank the management, Karpagam Academy of Higher Education, Coimbatore. Secondly, I'd like to thank Dr. A. Sangali Muthu, HOD, Department of Biotechnology. And I'd like to thank Dr. A. Arun Kumar, the Organizing Secretary, for giving me opportunity to present my paper. I'm Raj Kumar. I'm a research scholar. I'm doing my PhD at Tanjavur, as well as I'm an assistant professor working at Manipur State. It is in the northeast India. So today I like to present characterization of phytoconstituents and the antioxidant property of Reishi tinctoria. And today the outline of the presentation is first I will give introduction of this medicinal plant. Second, sample collection and preparation. Third, what are the methods I have followed? And fourth, I will tell about result and conclusions. Let me go first, the introduction of this particular medicinal plant. Reishi tinctoria is a flowering plant species. And second point, we can find this kind of medicinal plant in India, Nepal, Australia, Myanmar, and Vietnam. And especially we cannot find this kind of medicinal plant in the hilly stations. We can find only where there is dry and moist region. Even I have collected the plant sample, uh, Myanmar country, that is near to Manipur state. And Myanmar, we call them Burma. The old name, we call it Burma. Now, we can find only, as I told you, we can find this kind of medicinal plant in dry and moist region. Now, these plants, why we call it, this is a medicinal plant, we can use all the part of the plants. We can use it flowers. We can use that flowers as a medicine, leaves as a medicine. The seeds can be used as a medicine. Part that is also can be used as a medicine. And next, when we talk about the size of the plant, that we call it small to medium size tree. We call it as a tree type. It can grow up to 10 to 49 feet. You can find the tree even 10 feet, and you can find the highest peak will be 50 feet or 49 feet. And when we look at this part, we call it in Tamil pattai, mara pattai. Uh, it is very smooth, yellowish and brown color. And when we talk about the thickness, it is 10 mm thick. And when you look at the sample, as I told you, we can use all kind of the part of the plants. We can use the root, we can use stem, we can use bark, we can use leaves as a medicinal plant. And when we talk about these leaves, it is very simple. And the long, the distance, 10 to 20 centimeter long, and the width it is only 5 centimeter. Now, even it is available in Tamil Nadu, especially in dry area, Dharmaburi, even Tanjavur. I have collected the samples. And this plant, once we cultivate today, it can flowering only after 5 to 8 years. Today you are doing plantation, and within 1 year you won't get the flowering. You have to wait another 5 to 8 years. Now when you talk about the plants, what are the phytoconstituents are present? I mean, what are the phytochemicals are present? By characterizing the phytochemicals or isolation of this phytochemical from this particular plant, what are the uses? As I told you, it is a medicinal plant. It can able to cure in jaundice. If you go to villages of different Tamil Nadu state, they are using this one as a jaundice purpose. Those are infected, those are having disease with the jaundice. And that extract of leaves, they will put on the eyes, the drops. Even those are diabetic, they consume this one, the leaves and the bark. And those are infection on the skin. The leaves extract can be applied and they can able to cure the fungal infection. And antibacterial activities. And also have sometimes having infection by the bacteria, they can consume it. And coming to the main point, antioxidant. An antioxidant means there are some free radicals. I mean, those are smoking and drinking. The body can able to produce free radicals, and the free radicals can damage the cells. We have made up of cells. Now, when the cell is damaged, there is possibility of getting cancer. Now, by consuming this medicinal plant, it acts as an antioxidant. It can react against the free radicals. That's the main topic of today this presentation. And it is recognized as an effective therapeutic plants. There are a number of plants in India. They are using at a village level, but it is not recognized. But this Russian tinctoria, it is a recognized effective therapeutic plants. In Tamil language, we call it, even after this presentation, you may go and talk to your elders, the grandfathers and grandmothers. They will know the plant names. We call it Vepalai, Palai, Niram Malai, Nilapalai. That is the local name of the Tamil language. Now, I have given the introduction. Moving to the next, sample collection and preparation. How the samples are collected? We have to, I have plucked the fresh leaves from the Tanjavur area. So while we are plucking, you know very well, you have to wear the gloves and you have to pluck it. And second point, what I have done, you have to wash with the distilled water, not running water, because the distilled water is free from microbes and free from minerals. And third point, I have dried at room temperature. I should not keep this plant in hot oven. I should keep at room temperature. And after then what I have to do, what I have done, I made it powder form by using grinder or maybe mixie. Make it powder. Then after making powder, I should not simply leave it. I should keep it in the container in the closed conditions. Then further, we can use this sample to proceed uh, my work. I have proceeded with my work. So from there, I have taken 10 gram of powder mixed with 50 ml of solvent. Here I have used methanol. And what are the methods I have followed here? I have followed 4 methods to find out the phytochemicals and antioxidant property of this medicinal plant. So I have used gas chromatography, mass spectrophotometry, We call it in simple GCMS analysis. By using this method, I have found a lot of phytochemicals from this plant. And second method and second test what I have used, Fourier transform infrared spectroscopy. We call it FTIR. And by using this method also, I have found out a lot of phytochemicals which will be helping us in various purposes. And third methods, hydrogen peroxide radical scavenging activity assay. And through this method, I found the antioxidant property of this Reishi tinctoria. And fourth method, I have used thiocarbitric acid (TPA), and through this method, I found out a lot of the property of antioxidant of this particular plant. Now, I have given the introductions and the sample collections and what are the methods I have followed. Come to this result and discussions. You can see here the table which is smaller. The table number 1 is showing that the phytochemical constant in leaf extract of Reishi tinctoria by using gas chromatography, mass spectrophotometry methods. So here I have found a lot of components: neophytodiene, erythrose 78 bromo chlorospatula. There are a lot of components, lot of phytocomponents that will be helping us. And another next table, by using the fluorine infrared analysis, the second method. By using this method, I have found out a lot of phytochemicals which will help us in a lot of ways. And third one is I have showing the graph sheet. It's coming slowly here. The antioxidant property, as I told you, antioxidant means the lot of our cells to stop the damage by eating up the leaf extract or maybe that flowers or bark. It can able to inhibit the free radicals. Will not allow the free radicals to damage the cells. You can see the percentage of the inhibitions. First one, the green color, thiobarbitric acid, the inhibition level is almost 10 to 50% of inhibition will be there. May not able to cure completely, but it can help us little bit. That's about the percentage of inhibition by the Reishi tinctoria act as an antioxidant. And finally, what I came to the conclusion by following this number of methods, Reishi tinctoria leaf extract has variety of powerful bioactive chemicals, and I have found out phenols, amines, alkanes, carboxylic acid, aliphatic compounds, carbonyl compounds, and esters. These are the phytochemicals which will help us in a lot of ways. It can be act as antibacterial, antifungal, a lot of things. Not only that, Reishi tinctoria leaf extract also found the highest value of antioxidant properties. And if you keep on consuming maybe monthly once, maybe monthly once till your lifetime, there is less possibility of getting cancer because the cancer will be caused by the free radicals. The free radical will be produced by our lifestyle. Some will be keep on smoking, some will be keep on drinking. So because of that, the body can able to produce the free radicals, which will cause cancer. So this is my presentation, and I'd like to request everyone if there is any doubt, you may ask me. And thank you once again, the Karpagam Institute of Coimbatore, and Dr. A. Sangalinguta, the HOD, and Dr. A. Arun Kumar, the organizing secretary, giving me this opportunity to present my paper. Thank you.
Thank you, Mr. Rajkumar. Stay back. If anyone have any questions, you can ask.
Sir?
It was a good presentation, Mr. Rajkumar, and I could find a few flaws with the slides. First of all,
Yes, sir.
I will find the scientific names are not in italics.
Scientific name?
It's not in italics.
Okay, sir. Okay. It is because my presentation is supposed to be tomorrow, sir. But since I have some work tomorrow, so I made a little bit hurried. Okay, sir. I will change it.
Okay. And the next point, you have
Yes, sir.
analyzed mass spectrometry. Why have you not used the
Yes, sir.
HPLC?
Sir?
You have used the GCMS, right, for phytoparticulates.
Yes, sir.
Why not
Yes, sir.
HPLC?
Why can't?
Why not you have used HPLC?
No, sir. Actually, that's a good idea. Uh, actually, I'm doing my project work in Teripam. Hello.
Okay.
Actually, I'm currently doing my PhD because there are number of methods are there. I have not displayed all the methods. So it is my second year PhD. This year I'm doing this four methods. Next year I will be following another four methods. I think there is HPLC also there is high performance liquid chromatography there is. I think next year I will do after this second year over.
So in GCMS, how many constituent components you have identified?
Around 8 components, sir.
So what are the major components which have high activity?
Now, I think I have shown in the slides. There is phenol components,
Can you show me the
uh,
components which have high activity?
amines. Yeah, I will show you slides, sir. Amines. I have found out alkanes, carboxylic acid, aliphatic compounds, carbonyl compounds. That's what we found out. Here, sir. You can see the slide now. Here, this network is a little slow. That's why.
And I have one small another question. The sample which
Yes, sir.
you have collected, what is the size of the sample?
What is that?
Size of the sample, sample size.
Plant size.
The number of sample you counts. How many samples have you collected for the extraction of?
I have collected two. Yes, sir. I have collected sample at Tanjore as well as near Manipur. There is a Myanmar. We call it Burma in old names. And in the village in Manipur, there is one border state is there, More, and full of Tamilians are living there. Around 30,000 people are living there. Through the permission of the government of Manipur, I have collected sample even from the Myanmar. Call it Burma.
Okay, thank you, Rajkumar. Thank you for your presentation.
Thank you, sir.
Next, Ashwini, are you on the line?
Yes, sir. Yes, sir.
Can you able to present now?
Yes, I can present now.
Okay.
One second, I'll share my screen.
Okay. Present your screen. If there any all is okay, we go with the live.
Can somebody confirm me whether you can see my slides?
Yeah. It's clearly is visible.
Oh, oh, thank you so much.
Yeah, go with your presentation.
Good afternoon, everyone present here in the venue, and everyone organizers and HOD of the department and everyone. So today I am R.K. Harshvardhani from Commerce College of Engineering and Technology from Department of Biotechnology. Today I'm going to present my topic on analysis of protein phosphatase, PP2C gene, which is present in sugarcane. So what are PP2C genes? The PP2C genes are a gene that plays a very important role in the phytohormones, developmental processes, and diverse stress in most of the organisms, particularly in plants. So these PP2C genes are a sign of evolution, and they are located in archaea, fungi, bacteria, animals, and plants. Since they are present in all of these animals, it shows the importance of this particular gene. So why uh, we have to do analysis of this gene? That is the question I'm going to answer here. Because in in sugarcane in other plants, it is found that drought stress or abiotic stresses like cold and drought oxidative stresses has a major impact on the yield of the particular plant. So what in for that we need abscisic acid. Abscisic acid is produced when plant undergoes that particular type of stress. In this, as in this slide, you can see that ABA mediated abiotic stress response. So it is shown here clearly that the negative feedback of this TP2C genes provides the increased abscisic acid so that the plant may overcome the drought stress or any other stress, environmental stress or humanly induced stress. So PP2C plays this an important role. But why choose this gene? Because in sugarcane, as it is not a monoploid, and the analysis of this particular gene is not the technology is not developed. We we have to analyze first in silico and then go from the wet lab. So first what I did, my objective was to do a genome-wide analysis of PP2C genes in sugarcane and identify what are the genes, uh, what identify the PP2C genes that are present in the sugarcane. The next one, what are the materials and methods I use? The resources here I used are the databases like NCBI, Sugarcane Genome Hub, Phytosome, and Ensemble plants. These, to identify the PP2C genes present, particularly in the sugarcane, I used the Sugarcane Genome Hub. And to identify the similar sequences from sorghum and sorghum bicolor and Oryza sativa japonica thing, I used NCBA, Phytosome, and Ensemble plants. So there are articles that suggest that Oryza, uh, rice, and sorghum have PPTC genes, and they have done research on that, but not in sugarcane. So using those as references, I have followed the methods. So here, uh, first I identified the PP2C genes using the databases, and to confirm that these genes were PP2C genes, I did KEGG analysis, motif analysis, and genes motif analysis, and whether they perform the phosphorylation or not, potential phosphorylation sites. I did net phosphorylation analysis, and to identify the protein sequences and the ORF present in this particular gene, I used ORF prediction tool that is offered by NCBI. So from there, I was able to identify the CDS sequences, ORF sequences, and as well as the protein sequences. And then I also did a gene structure analysis as I have shown in the third step. Gene structure analysis and protein structure analysis I have performed to identify the secondary structure of particular protein and all. And then the last one was to identify target sequences and produce oligos for that target sequences. This was performed by using CRISP-OR, which is a free technology available, which is a free tool available. From there, I was able to identify uh, the target sequences and to prepare potential oligos for that. So The next will be here. I have shown some of the home pages of the particular databases. Sugarcane Genome Hub is a database that provides the genes, mRNA sequences, genome sequences, and the CDS sequences, and other chromosomal sequences of all those related to uh, of R570 cultivar and phytosome. Most of the plants genome sequences are present in that. Use for sorghum bicolor and Oryza japonica. I used both the phytosome as well as the ensemble plants. As you can see, I used the ensemble plants for uh, uh, for identifying the PP2C genes. They were used as the reference sequences, and I used ORF Finder tool, as you can see the home page for this. And in the results, I was able to totally identify 671 sequences that are that has the property of PP2C genes, and only I select finally 69 genes were selected because of their length and their blast properties and e-values and all those things. 69 I was able to identify. So here I have given the information of what are the PPT, what are the gene, particularly mRNA sequences name, and their description and their length and position in which chromosome they have located and all that. So the length range from nearly 900 to 3000, 3500 sequence base pairs. And after the identifying the total 69 sequences, I performed the ORF identification. For ORF identification, I concluded that the sequences had 77 base pairs to 958 base pairs of CDS present in it, and smaller nucleotide sequences and protein sequences were neglected for the further analysis. So here I have given a schematic representation of the intron-exon structure of the PP2C genes in the sugarcane. As you can see, nearly three in exons to nearly 12, 13 exons were I was able to identify in all those sequences. I have given the results for all the 69 sequences. Here, what tool I used was gene structural display server. The tool from that tool, I was able to identify the structure of the genes. So here the violet color represents the upstream and downstream UTRs, and the yellow color describes hello, am I audible?
Yeah, Harshvardhani, you are audible. Continue your presentation.
Thank you. So the next one is I've done this gene structure display server, and it is important because the longer the CDS, the proteins that produces will be more. And I'm sorry for the interruptions from the messages, since it was an immediate presentation. So the next thing I was able to do was secondary structure prediction of the proteins. So by predicting the secondary structure, after predicting only the secondary structure, I was able to move to the 3D structure. And I'm in the process of doing the 3D structure prediction. But here you can see mostly the proteins that have the protein phosphatase 2C uh, as their motif had alpha helix and random coil more. Then the extended strand and the beta strand, they were present in very low number. And then so that here it shows that alpha helix of the protein structure is more prominent in the PP2C genes, and followed by the random coil, followed by the followed by the extended strand, and followed by the beta strand. So I have given all the information about the 69 sequences. here. And the next will be the motif sites present in the PP2C genes. I was able to identify 20 motif sites, which was identified using mean suit, which is a motif finder tool, and minimum 10 to maximum 20 motifs were present in it. So it confirms the presence of conserved regions in it. So since conserved regions were identified and motifs were identified, I was able to confirm that this particular this particular protein performs the function of the PP2C genes. So here I have given all the 69 sequences motifs. As you can see, the whole protein sequences has the property of having a conserved region. And so next is followed by the CRISPR results. After confirming that these are the TP2C genes, I did find the target sequences. There were 332 possible guide sequences. It was identified in the CRISPR results. As you can see, these are the results of the CRISPR. It is a graphical representation where the PAM sequences start and end. So here the PAM sites and the expected cleavage positions were located, and it is shown clearly that the green indicates the high specificity, and yellow indicates the medium specificity, and the red indicates the very low specificity. So here I have given the graphical representation of those CRISPR results, and as you can see, there were 332 possible guide sequences. From that, only 2 sequences were selected, and it is shown in next slide. So here I have selected these 2 PAM sequences because they were particularly prominent to BS1, the BS1 enzyme, and the gene ID I was using for that particular is SH. I've given the name of the gene ID and the length and the exon's length. And why I chose this particular gene ID was because it was downregulated. Nearly the log4 to change value was -3, which shows that it is very low downregulated. As much the downregulate as per the most downregulated genes, the in the particular ABA signaling pathway, it shows that it is most prominent. It is the most prominent negative feedback regulator. So that is why I chose this particular gene. And you can see that the forward and reverse complement, and I have shown the location in which the particular gene has been present, in where the target sequences are present. So the next one will be the potential phosphorylation sites in PP2C genes. Here you can see serine is present in most largest quantity, and followed by threonine and followed by tyrosine. So this proves that the gene performs the function of phosphorylation, and all the genes were performing phosphorylation, and potential phosphorylation sites were identified here. And it is followed by gig analysis. This is what proves that the PPTC genes is actually present in it, and what type of pathway it takes. It performs its role. So here you can see there are five and six different types of PP2Cs here present. So one is pyruvate dehydrogenase phosphatase and protein phosphatase 2C, and protein phosphatase PTC23, and protein phosphatase and PP2CG and PPM1G, protein phosphatase 1, and PPM1L, which is protein phosphatase 1L, and PPTC2, which is if both the PP2C and PPTC are similar and same. So it has shown most of these these seven were present in every sequences randomly. So I was able to identify it. So next one will be doing a after doing the Kig analysis, I have to do the gene ontology so that I can confirm that these perform the particular function, and after that I'll be performing expression studies, phylogeny analysis, and all this. These will be in my future work. And I would like to thank uh, Director of ICAR SBI, Shurikan Breeding Institute. And I would also like to thank my guide, R. Manimegalai, and Ronaldo Anu from Commerce College of Engineering and Technology for providing this opportunity. And I would like to thank the organizers of this national conference from Karpagam College of Engineering, Karpagam College. And thank you for this opportunity. If you have any doubts, you can ask me.
If you have any doubts, you can ask.
Thank you, ma'am. That was a nice presentation. It was really great. You see, we have sequence around 300 pieces, this was guidance sequences, right? Am I audible?
Hello? Excuse me?
Am I audible?
I'm not able to hear anything. Can I stop the presentation?
Am I audible now? Can you hear me?
Yes, you are. Yes, you are audible.
That was a nice presentation and it was very elaborate. Thank you for your presentation. And
Thank you.
we had around 336 guideline sequences in CRISPR, right?
Yes.
Have you tried anything or do you have any plans of trying it out?
Yes, we have synthesized the oligos, and I have chosen this particular two sequences, and we have synthesized the oligos. And the wet lab results have not yet been released, so
So you're waiting for the results then.
yes, I'm waiting for this.
That was a nice presentation. Thank you.
Thank you so much.
Thank you, Harshvardhan.
Yes, stop the presentation. Thank you.
So thank you every participants who have stayed in the Google meeting for last. So thank you all. Have a nice day.